Hello, can you hear me in the back? Yes, good. So my name is Mira. I'm an associate professor in the School of Life Sciences at UNLV. And thanks to Allison and Frank, I'm very happy to be given an opportunity to share our work. The title is Learning the Effect of 3D Genome Organization on Gene Expression. And I'm going to divide the talk into two parts. So the first part, I'm going to do a short review on what we learned in the last 10 years or so about long-range protein interactions with high-C data. And I'm going to use an example of the Hox gene clusters. So the first part of the talk is all other people's work. It's not going to be our lab's work. And then in the second part of the talk, I'm going to introduce our lab's approach to use machine learning to understand whether these long-range chromatin interactions between the enhancers and the target gene affects whether there is a functional relationship between the enhancer and the gene or not. So let's start with the Hox genes. So I thought this example would speak to the audience that we have here, because a lot of people are interested in evolution and development. And the Hox genes are famous because they're the master regulators of embryo development that determines which segment becomes which body part. And it turns out that these clusters of genes are shared and conserved across all bilateral animals. And they are also very interesting for people who are interested in genome evolution because they tend to have a very conserved position in the genome. So we have these, the vertebrates that went through 2 rounds of whole genome duplication that have 4 copies of these clusters on different chromosomes, and they all have conserved their orientation and order and the cluster on all 4 chromosomes. And it seems like they have some meaning because if you look at which genes are expressed, they tend to be expressed in an order temporally from early development to late development. And they are expressed in an order also physically from the anterior part of the body to the posterior part of the body, so that the location along the genome corresponds to the temporal order, and it also corresponds to the order along the axes of the body. And this was known for a long time. Recently, what was discovered was that this regulated expression in order along these genes, along the chromosome, is also aligned with the methylation and demethylation that happens along the chromosomes. So yesterday, we talked about epigenetics and the modifications on these histones that the DNA is wrapping around that will change the chromatin structure of the chromosome and change whether that region is allowed to express or not. And it turns out that epigenetic modification also goes through this change where originally it's all methylated, and that methylation is maintained. But then you go through modifications as time goes by and the organism develops, and the chromosome opens up so that the first gene, the chromatin is modified first, and then it gets expressed first, and then the later genes, gene 13, gets modified later, and then it gets expressed later. And in addition to that, even more recently, we learned that this change in epigenetics also comes with change in the overall 3D structure of the chromosome. So if you look at the nucleus and look at how the chromosomes are structured, you see that the structure of the chromosome also opens up and changes from a more compact silent form into a more open, accessible form. And how do we know that these chromosomes are changing? Initially, they were observed through these observations on the microscope where you're actually looking at the chromosome. But nowadays, they can use these new techniques that can measure these long-range chromatin structures in genome scale way. So this technique is called Hi-C. And the way it works is, so these are the DNAs, and the DNAs are wrapped around proteins usually. So the most common protein around here is going to be the histones, but sometimes you'll have some transcription factors as well. And scientists use the formaldehyde to fix these proteins with the DNA. And when it does that, any DNA that is next to it will be fixed together. And then you cut the DNA into pieces and tag them with certain marks. And then you use ligating enzymes to connect them together. So initially, this DNA is part of different parts of the chromosomes, but they're just physically close to each other. But now they are connected. And then further fragmentation occurs, and you pull down by these markers, and then you can sequence this DNA, and you're going to get part of the DNA coming from this blue part of the chromosome, and the orange part of the DNA is going to come from this orange part of the chromosome. So although linearly they could be very far apart, if they're physically close together in 3D, they're going to be captured together. And then we use these sequencing technologies to read what these DNAs are that are connecting each other, so that you get these reads, the orange reads representing the orange chromosome part, the blue reads representing the blue chromosome parts. And then we use the sequence similarity to figure out what part of the genome these reads are coming from. And you can count how many reads are coming from which part of the chromosome that are connected between the orange and the blue parts. And through that kind of counting process, now you can measure the proximity between the chromosome parts. Because when they first published this method that was called Hi-C, they were able to show, although it's you can't really read this right here, what they were able to show is that they could measure the distance in the microscope, under the microscope, using a technique called FISH, and measure that physical distance, and then measure the number of reads that are paired between the locations, and they saw a very high correlation between, very high negative correlation between how many reads are together and the distance is apart, so that the number of reads can be a proxy for measuring proximity in 3D space. So then you get these kind of graphs. So we saw these graphs yesterday as well. And along this diagonal is your chromosome. So you can see that the intensity is the highest along the chromosome because very close to each other touches more often, right? So anything that are close to each other is going to show up as very dark color. But then you get these corner dots, we call them corner dots, where along the axes of the chromosome, they're quite far apart. So for example, this one right here, you're in the middle of the chromosome, and this end is at the end of the chromosome. But then between the two positions, you get this intensity right here that shows up as a red dot. And that tells us that there were reads that were produced that map to this position of the chromosome and this position of the chromosome. So that kind of signal can tell us that there is interaction between very distal parts of the chromosome. And by looking at these kind of figures, they were able to find that these chromosomes tend to have these blocks of boundaries that show a more higher frequency of connection or interaction within these blocks, but then there are boundaries such that you don't get as many interactions between positions along this block and position along this block. So you get these, what people call domains. And these domains have nested structures so that you have a very light colored square, but then within a square you have a more darker colored square. And that gives you this kind of nested domains where these parts of the chromosomes are all interacting with each other, but within this domain you have a subdomain that is even higher intensity of interaction going on. And these kind of domains were named topologically associating domains, or tags. And tags show boundaries on that high C graph. That's how they're defined initially. And it means that the DNA sequences within the tags interact with each other more frequently than with DNA outside of tags. And the corner dots that show up as intensity off diagonal, those dots show that the pairwise interaction is more frequent. And they were able to do this high C and apply it to different genomes. The human was the first, but then they applied it to other species. And what they found was that these structures tend to be very conserved, and especially the larger structures are more conserved. But then if you go look at the smaller structures within the domains, they are more variable, and they're variable across species, but they're also variable across tissues of the same species. So if you look at embryo as it develops, or if you look at liver tissue versus blood tissue, you're going to find that overall large-scale structure is still the same, but the small loops tend to appear and disappear. So going back to the Hawks example, we know that as the genes get expressed temporally in order, the epigenetics change and also the genome structure change, but does that have a causal relationship? Is the genome structure change causing the gene expression to change, or is it just a coincidence? You have to express these genes, so somehow the structure gets changed. So that was the main question that drive the field when they were first discovered. Does the form follow function, or does the function follow form? Do these tags, the genome structure, have any kind of functional role in the regulated gene expression? And there were two opposite observations that were going on. There were observations that told us that yes, these tags create insulated neighborhoods where the regulatory elements that are interacting with the promoter of the genes that have to get expressed are bounded by that structure, so that if the regulatory element is within the tag, you have more frequency of promoting or activating the gene. If the element is outside the tag, then you are insulated from activating the gene. But there were also other observations that seemed to suggest that no, we go and disrupt the tags and we try to change the structure artificially, but we don't see that much gene expression change. What's going on? And there were also some other observations that suggested that the causal direction is the opposite. So sometimes the polymerases that are transcribing the gene, the elongation process, seems to change the structure of the chromosome. So maybe it's not always structure dictating gene expression, maybe gene expression can also change the structure. So it's a very confusing state right now. And there are many different observations that tell us confusing stories. But some of the evidence that are supporting the functional role of tags are studies that looked at how artificial regulatory elements introduced into the genome drive the expression of the genes. So scientists have introduced artificial enhancers with a reporter attached to it into different parts of tags along the chromosomes and looked at how these reporters are expressed along the embryo, the pattern of the expression. And what they found was that generally the random insertions that are within a tag tends to show correlated expression patterns, but then the random insertions that go outside the tag, they tend to differ in the expression pattern. So those kind of observations seem to suggest that the boundaries seem to matter in terms of expression of the genes. Also, there have been series of studies that disrupted the chromatin structure many different ways. So here are only 3 of them, but there are many more studies. Either they deleted certain regions that demarcate the boundaries. So these boundaries, what people have found is that the mechanism that creates these boundaries tend to be associated with a certain protein called CTCF. So if you have CTCF binding that in a certain direction, they create boundaries. So what if we remove the CTCF motif so that the CTCF cannot bind anymore? That will merge the 2 independent TAs into one. How does that change the gene expression pattern? And they found that indeed that will change the expression so that now previously inaccessible enhancer can activate a gene that was previously not activated before, and that can lead to developmental abnormalities. And the same was similar experiments were done with inversions, and they also saw gene expression change and developmental change as well. So it seems like there were enough studies that seem to tell us that at least there is something going on here. But then the alternative theories were supported by other evidence, like this study that was done in 2019, where they looked at actual physical chromosomal distance under a microscope and tried to see if the transcriptional bursting status correlates with the 3D distance between the enhancer and the promoter. And what they found was that transcriptional burst, when the gene actually gets transcribed, didn't show very much correlation with whether the enhancer was physically close to the promoter or not. So that seemed to suggest that physical proximity in 3D space is not as important. Another study that was showing a different evidence was instead of trying to go in and manipulate the genome itself and try to change structure that way, let's just deplete the proteins that construct these genome structures, like the CTCFs or the cohesins, and just disrupt structure in a genome-wide scale and see how much gene expression changes. And when they did that, they basically depleted cohesins, which are one of the proteins that construct these structures. After, I think it was after 6 hours, they only saw 64 genes change in gene expression. So that was such a modest change after such a drastic disruption of structure that they concluded that structure must not have a strong effect on gene expression in a genome-wide scale. Another study was done later in an independent lab, and this time they waited longer. They waited 5 days after depleting all the cohesins. And that after 5 days, they saw about 1,000 genes change gene expression. But then they couldn't conclude whether this 1,000 genes changing gene expression was because of the direct causal effect of the structure change, or because of these early genes changing expression and having indirect effects within the 5 days. So as you can see, it's a complex topic, and there are still discussions going on in the field. But as more and more studies are done, we are growing support for the hypothesis that these structures does seem to have some effect on regulation of the genes within those domains. And although disrupting a domain will not always lead to gene expression change, the relationship is not that straightforward, but we have enough of the evidence where it does happen so that we know there is some role in regulation. So going back to the Hox genes, so these clusters, they are well known for the early trunk axes development, but they have been co-opted many, many times for development of other body parts as well. So one of the other example is after they form the overall body axes, they are also used for limb development so that you express different copies of the Hox genes as you try to develop the arms and all the way to the fingers. And when this is occurring, there are two different waves of expression going on. So when the early limb is developing, you have the early Hox genes express more, and the late Hox genes are suppressed. But then as the limb grows and the digits, distal part of the arm are developing, you have, there's supposed to be an opposite triangle down here. You have the later Hox genes increase expression and the earlier Hox genes get suppressed. So these two different waves of expression have been studied, and they know that they're regulated by different regulatory elements on either side of the cluster. So the early Hox genes are regulated by the 5' side of the cluster, and then the later Hox genes are regulated by the centromeric side of the cluster. And in 2013, scientists from the lab of Denis Duboul have been studying this cluster for a long time, and before this study, they had already identified the enhancers that are responsible for the later development that are in the centromeric region. And in this study, they went on and identified additional enhancers that are regulating the telomeric part of the cluster that are responsible for the early development, the arm and the forearm. So you can, this is showing you that initially you have the early Hox-D genes that are expressed, and then as development goes on, you get the later Hox genes expressed in the fingers. And this bar chart is showing you the expression pattern change. So the black bars are the early, and initially you can see that Hox 3, 4, 8, 9 are expressed more. And then the gray bars are the middle development, and the white bars are the late development, the digits. And the white bars show that the early Hox genes are suppressed, and now the later Hox genes, the 10, 11, 12, and 13 is shooting up. And it turns out that this regulation lies in two tag boundaries that are showing up right where the Hox gene cluster is. And within this, so the Hox genes are clustered around this region, and right next to the clusters are a gene desert, a long stretch of non-coding elements that only has these regulatory elements that drive the expression of the Hox-D cluster. And if you look at how the Hox-D genes are expressed, the early Hox-D genes, which are on the left, right side, are, so the Hox-D1, there's a label here that says Hox-D1. Hox-D1 is not expressed at all during limb development. So if you look at the expression, it doesn't show up. And if you look at the interaction, this is measuring something similar to high C, but in a local scale called 4C. You can see that the interaction between the Hox-D locus and the regulatory region is only to the left, right? only to the centromeric side. And if you look at the other end of the HOXD cluster, HOXD13, the late development gene, they are expressed later during development, and they are regulated by the telomeric part of the regulatory region, these purple enhancers. And you can see that the interaction between the cluster also only happens on the left side. So if there is an enhancer in this region, the HOXD13 only touches the left side of the enhancer. But what about the genes that are in the middle? It turns out that the genes in the middle, they switch the contact, which domain it interacts with as the development goes by. So you have the HOXD9, which is initially expressed early in the limb development, and it contacts with the enhancers on the right side early. And it's the same with the HOXD11. It's connecting with the right side of the enhancers. But as the development goes by and you get these purple digits, genes, the later HOX genes expressed, you can see that the middle HOX genes now start to interact with the telomeric enhancers. So it shows that it still didn't show whether this interaction change is causal to the regulation of this gene, but it is aligned with the expression change that is going on. So it shows that the contact of these middle genes initially happens with the domain that is active at that time. And as the domain gets suppressed and the alternative domain gets activated, the interaction now switches to the latter domain. So now the hypothesis is that there are these two domains, the centromeric domain, C-dome, and the telomeric domain, T-dome, that is driving the regulation of these HOXD clusters during limb development. And this was done in 2013, 2017. And this lab since has done a series of experiments trying to take out different parts of the genome and trying to show definitely that this change is driving the expression of the genes. And it turns out the story is never that simple. So the disruption studies that they did change the expression in time. So if you look at the development at day 9, the development is delayed. So the genes that are supposed to be expressed in the limb at day 9 is not expressed at the right time. But then if you go and wait until day 12, all the regular expression resumes. So it seems like these changes in structure can change the regulation of the gene in a subtle way, but that the program is robust enough that you still get normal development later on as time goes by. So maybe there is fitness effects in the long term, but they weren't able to measure it. Okay, so in summary, the genome is organized into nested structures that delineate the long-range interactions, and these domains are called TAS, topological associating domains. They define the boundaries of frequent DNA interaction, and they can contribute to gene regulation by restricting the space of enhancer and gene interaction. Disruption of TAS show conflicting results on the impact of gene expression, and the functional role of these structures seem to be subtle and complex. But evidence is accumulating for a relationship between 3D genome organization and its regulation. Now I'm going to switch gears into the work that our lab is doing. We have been interested in understanding the evolution of these regulatory elements that people call enhancers and where these novel cis-regulatory relationships arise from. And especially we're interested in transposable elements because transposable element integration seems to be a fertile ground for where these novel regulatory elements arise. We wanted to model how transposable element co-option occurs. And we needed a good model to identify functional enhancer target in pairs relationships. that can distinguish transposable elements that has been co-opted as an enhancer versus a transposable element that is suppressed and it's noise. So that was kind of where we're coming from. And then this project turned into a machine learning project. So we're kind of diverging from our original goal. But fortunately, nowadays, we have a lot of data that can model the relationship between enhancer and target gene regulation. So it comes from this new technology that combines CRISPR with single-cell RNA-seq. And this slide is copied from the company called 10x that will provide this kind of service for you. So how it works is you have these guide RNAs that you design for a CRISPR experiment to knock down whatever your target is. You can do that in a massively parallel scale. So you generate many guide RNAs, and then you transduce single cells so that it gets multiplexed into cells randomly. So every single cell has a guide RNA, or we can have multiple guide RNAs, but they are randomly distributed so that you have different guide RNAs going into different single cells. And then you do single cell RNA-seq so that you can read out the expression of each of these single cells. And here this is their control experiment that was done as a proof to show that it works. They generated guide RNAs for different genes, COA3, COX18, COX6C. So there were 2 guide RNAs for COX6C and a control. And then they look at the COX6C expression in these single cells. And what you see is that only the single cells that has this guide RNA for COX6C, you see the COX6C expression go down. So in principle, it seems to work. And you can then take all of these single cells and separate them out by what guide RNA they have inside, and then look at the overall expression of whatever target gene you're interested in within this cluster versus the rest. And that will tell you whether it has an effect or not. And these kind of approaches has been done so far in 3 different studies where they used CRISPR interference. So here you're not knocking out your target, you're just suppressing the target by binding a PRAG domain, and you're blocking the action of the enhancer. So they have done this, and the scales are very impressive. So in this first study by Fulco et al., they were able to target about 800 enhancers. And at this time, they weren't doing single-cell RNA-seq. What they were doing was they had reporters for different target genes, and they were able to cell sort them by their expression of the genes, and then quantify the gene expression that way. So that's why the gene targets were a little bit lower in scale. But by Gasparini et al., or a newer paper that was published in 2020, now you are using single-cell RNA-seq so that you don't have to do these reporters and cell sorting yourself. And now you can measure about 10,000 genes expression at the same time in each single cell. So this was the largest study done so far, where they targeted 5,700 enhancers and measured the readout of about 10,000 genes. And they were able to achieve this because they weren't trying to get one guide RNA into one cell. So it wasn't a one-to-one relationship. If they were to do that, the amount of single cell and the amount of reads that they had to generate would have been enormous. But instead, they decided to multiplex the guide RNAs so that each single cell has multiple guide RNAs. But then, as you separate them out by the guide RNA, you can still see the effect on the expression as long as there is a signal, even if there are other guide RNAs that could be inside these cells. So you take these cells, we know which guide RNA are acting in each cell, and then you take all the cells that have that versus the rest of the cells that don't, and compare the expression of the gene. And if you see a significant drop, then that means that the enhancer that you were blocking with that guide RNA has an effect on the gene expression. So this is the data set that we decided to use for our machine learning algorithm. But before we go there, let's just introduce what the existing model was. So after Fulco, the first study, generated this data set of 800 enhancers blocking and 30 genes expressed. Based on the observations from that data set, that they came up with this model that they call activity by content model. And it is a very simple model. The way it works in their model is that you have a gene, and this gene is derived by multiple enhancers. And the enhancer acts on the gene through this three-dimensional physical contact. The enhancer's relative strength depends on the activity that is measured by these epigenetic marks. And the marks that, there are many different marks that indicate enhancer activity, but the one that they use is H3K27 acetylation. And then another mark is what people call DHS, which is the overall accessibility of the chromatin. So overall accessibility and the H3K27 acetylation marks indicates how strong that enhancer is, and how strong the contact is. Those two factors determine the regulation between the enhancer and the gene. So you have two different examples here. So you have the black arrow, that is the gene, and then two different enhancers. The E1 has a relatively low H3K27 AC peak and is not as accessible, measured by DHS. Enhancer 2 is a stronger enhancer because it has a higher peak for the enhancer chromatin mark, and it is more accessible. But if you look at the contact, relative contact with the enhancer and the gene's promoter, you see that the high C, which is shown here, shows that E1 contacts the gene more frequently than E2. So although the enhancer is weaker, the contact is stronger in this case, so you multiply that and get a readout of 12. For E2, the enhancer itself is stronger, but the contact is weaker, so you get a 4. And because both of these enhancers are regulating the gene at the same time, for you to be able to detect one pair's regulatory relationship, that's going to be measured relative to the other enhancer's control of that gene. So you basically normalize it by the total number of enhancers that control the gene. So if you have an effect of 12 versus 4, then you measure it as 75% from E1, 25% from E2, something like that. So conceptually, they came up with this model based on what they know about how enhancers work in driving the promoter gene expression. And we were curious, since this model is all based on theory, whether a machine could learn the model better. And we thought about what kind of factors we could put into the machine in addition to enhancer strength and physical contact. And we know that when enhancers are activating a gene's expression, there are many other factors that are going on where many different proteins come and bind to the general transcription factor in addition to the RNA pole that drives the expression. So we thought that maybe if we know what transcription factors are binding to this enhancer locus and the promoter of the gene, that could give us more information on the relationship between the two. So these are the variables that we decided to put in addition to enhancer strength measured by H3K27 acetylation and high C. We decided to look at expression level of the target gene. This is not the change in expression level, which is our outcome. This is the overall just base level expression. The histone modification of the enhancer and the target promoter, transcription factor binding that we can gather from the study called ENCODE. And we also thought maybe these transcription factors are binding to the enhancer and the promoter at the same time. There's going to be a co-occurrence of peaks between the enhancer and the promoter of the same transcription factor, and that co-occurrence could provide us information. So those are the variables that we decided to put into this machine that will learn the model. So the input looks something like this, although it's not the whole table, where you have an enhancer locus that is defined by the chromosome start and end. You have a target gene. The target gene is always within 1 megabase of the enhancer. And we know the effect size and the p-value and the significance of that relationship from the Gasparini experiment that we just derived this data from. And we have these variables that will predict the relationship, the histone modification, enhancer mark, the chromatin accessibility, the one-dimensional genomic distance, the three-dimensional physical distance, ABC, which is the score that we just talked about, and the other transcription factor peaks that are measured from ENCODE. So this is just a summary of these variables. And the main question that we were interested in was, if we were to remove high-C contact information altogether from the model and just rely on genomic distance, can it still predict the functional relationship between the enhancer and the promoter? And or if we include the high-C contact information that will measure physical 3D proximity, will that predict it better? And the machine that we're going to use is called a decision tree. And there are many different machine learning models out there. This is one of the simpler ones. The way it works is you have these variables that you put in, right? The genomic distance, transcription factor binding, histone modification marks, and it tries to learn a threshold that will predict whether it's a functional relationship or not. So you do, is the genomic distance less than 190,000 bases? If it's yes, it goes this way. If it's no, it goes that way. Is the histone modification signal less than 17? If it is yes, it goes this way. If it goes no, it goes that way. Is it binding SP1 in the transcription start site? Yes, no. So it's very similar to if you were to draw decision-making process in your head for whatever decision you're making, right? The machine is making the same decision. But the reason why it is effective is that it doesn't try to learn one tree. It tries to learn many, many trees. And we call this ensemble method. It tries to learn many weak trees. Weak trees meaning that it's only a little bit better at making the decision than your random choice. So you make hundreds of these trees that are, each one of them are only slightly better at making the decision than your random choice. And then you draw the consensus from these 100 trees. And it turns out doing it that way gives you better predictability or predictive power for whatever classification you're trying to make. And this overall method is called gradient boost or gradient boosting decision trees. And it is one of the more interpretable machine learning methods out there. So we're going to use this method to learn and answer this question, whether 3D contact information is important in predicting which enhancer gene pair is functional or not. And it turns out, yes, having the 3D contact information improves the prediction of the true enhancer target gene pairs. So this is showing you just putting in the enhancer strength, the H3K27AC and the accessibility with the genomic distance. And this is showing you the overall accuracy that is measured with this measure called the precision recall area under the curve. And if you add to those enhancer strength and genomic distance, add to that high C contact, it increases the predictability significantly. And it's the same even with transcription factors. So if you put in transcription factor peaks, overall you can see that the prediction improves a lot, but having high C contact improves the prediction a little bit more. And if you put in co-binding peaks, co-occurring peaks between the enhancer and the promoter, you can see that again, you see an increase in the prediction power if you have the 3D physical contact information there compared to just relying on the genomic distance. Overall, what we found was that if you have transcription factor information, it increases the prediction more than not having the transcription factor information, but having co-occurring peaks between enhancer and promoter doesn't really increase the prediction very much. So seems like that doesn't really matter. And from these machines, you can infer which variables are important in determining which enhancer promoter pair is functional or not. And usually when this kind of tree is learned, the variable that comes early in the tree are the more important ones. So for example, in this tree, high C contact, is the high C contact value less than a certain value or not, is the first variable it looks at to make the decision. And you can measure that importance and rank the variables by their importance. And we ran this 10 times, and what you find is that high C contact is almost always in the top ranking of how important it is in predicting enhancer promoter relationship. And other important predictive features turn out to be the transcription factors that are well known to be regulating either, because this data set is based on the K562 cell line, so it's the blood cancer cell line. So these transcription factors are well known to be playing a role in both blood development and blood cancer. So those are kind of what you expect. But then you get also certain genes that show up very high in the ranking that you don't really expect. So now we're going to look at whether that model that Fulco et al. Derived from their data is a good working model of enhancer function by including that variable into the model. So if you add that ABC variable that combines, just to remind you, it combines the strength of the enhancer measured by the enhancer marked peak multiplied by the contact frequency. So that's their model. And if you add that score into the model, it increases the predictability quite a bit. So without it, we were looking at a precision recall of around 30. And then now it's going up to 35. So overall it increased. And still, high C contact is still very relevant. So if you have high C contact, it's always better than not having high C contact. And you look at the variable importance again, you see that ABC score is now that we included ABC score, it's always in the top of the list among all the runs that we did. And if you look at where high C contact went, since ABC score and high C contact are very related, they're correlated with each other. Once you introduce ABC score, high C contact drops down in the ranking. So if you have ABC score, then you don't need high C contact information as much. And this is the overall ranking summarized across the runs that we did. The best predicting variable is the ABC score. High C contact is the second, although here it looked like it wasn't really relevant, but if you add it up across all the trees, it turns out it is the second place. And then the third is distance. And then the fourth is this gene that is binding to the transcription start site that is called C11 or F30. We're going to talk about that soon. STAT5 transcription factor binding to the enhancer, and then other variables. And this is showing you the direction of the variable's effect. So ABC score, if your ABC score is high, that means you're more likely to be a true enhancer gene target relationship, right? An enhancer that when it's perturbed, it changes the gene expression. So this is the way to read this graph is it's binning all of your data into these bins by the level of the ABC score. And then the highest ABC score, all the rest of them, there's not very much change in the prediction value, but the highest ABC score increases the prediction, positive prediction. And that tells us that the direction is positive. High C contact is also has a positive relationship. If you have higher 3D contact, then you're more likely to be a functional relationship. Genomic distance is negative. Here, the pattern is opposite. The smallest distance has a higher prediction, right? So that means that distance and prediction has a negative relationship. What is this gene, C11 or F30, that shows up fourth in the ranking? This one has a negative relationship between the prediction. So if you bind this protein in the transcription start site, then you're less likely to be in that functional enhancer gene target pair. And it turns out that this gene has a name, although in this data set it was called something different. Its name is MZ. And it was originally reported as a transcriptional repressor that is in a part of a protein complex that also contains KDM5A, which is a histone lysine demethylase, for this activating methylation that is usually found in promoters. And this protein complex basically demethylases H3K4 trimethylation and suppresses the gene expression. So it's a transcriptional repressor. So that makes sense that when you have this gene bound to the promoter, it doesn't respond to the enhancer blocking. So to summarize, machine learning on this CRISPR interference enhancer perturbation data set suggests that 3D contact is one of the most important variables that can distinguish truly functional enhancer target gene pairs from pairs with no effect. And the activity by contact model, it reflects the data very well. If you include it, it's going to be the single best, most predictable variable. Information on transcription factor binding improves the prediction, and it can suggest whether it's a repressive or activating effect. Co-occupancy of the same transcription factor between the enhancer and the promoter doesn't seem to inform whether the enhancer target gene pair is going to be functional or not. Okay, so that's the work so far. But if you actually go look at the predictive power of these models that we learned, it's not very impressive. So this is showing you what the data breakdown looks like. So this is the actual effect, whether it has an effect or doesn't have an effect. And this is a very, very imbalanced data set. So they tested about, what is it? So this is 1/4 of the data. They tested about 60,000 enhancer gene pairs and they found 600 positives. So that's 1% positive rate, right? You're testing 100 and you're going to find one truly functional enhancer promoter pair. So predicting that kind of 1% is very difficult. And you can see that because majority of your data is negative, it's good at separating out the negatives. So if it's a negative, then you're likely to predict it as negative. But distinguishing among the positives are more difficult. So it has an effect, but we only predict 60 of them as true positive, and we still predict 80 as false negative. So it has an effect, but we predict it as negative. And as we're predicting the 60 true positive, we also predict 100 false positives as well. So if you look at what these categories of enhancer gene pairs looks like, you can see why this happens. Because you look at these misclassified cases where we predict it as functional, but there's no effect, you can see they look very similar to the true positives in terms of distance, right? They're very close by in their genomic distance, but in the experiment, it didn't have an effect. The false negatives, the ones that we missed, but it actually had an effect in the experiment, they look more like the negatives, true negatives, in that their distance is much longer than our true positives. It's the same with the high C contact information. So if you look at the ones that we predicted wrongly, they have very high C contact. And it looks like because they're having this close proximity between the enhancer and promoter, we call that as it's a functional relationship, but actually in the experiments, there was no effect. But if you look at the ones that we missed, but where there was true effect, there's no very strong high C contact. Their contact level is much similar to the negative cases, right? So that tells us that although physical proximity is the leading predictor for the model that we have so far, which is modestly performing, there's more to the story. Is complicated, just like the experiments that the literature, if you go into the literature, the experiments are telling us conflicting stories, right? So there are enhancer target relationships that are true, but don't have 3D contact intensity. And there are enhancer promoter relationships that seem to be interacting, but there's no regulatory relationship. So I have two students who are working on this project, and Dylan is interested in these cases where enhancers could exert a regulatory effect on the target without direct physical relationship. So he's interested in secondary connections through the network of these 3D contacts, and he's looking at that aspect of the project. He actually has a machine learning model that had a better performance than the one that I showed you, but I haven't done justice for his model. And Corinne is looking at how we can cluster the variables that we put into the machine before we put it in, so that the machine has a better, easier learning to do. So that's called unsupervised learning, and she's looking at that aspect of the project. Okay, so that's it for today, and thank you for listening to me. And I'd like to thank the publications where we draw the data from. Yes.
I was really intrigued by your analysis of the false and positives at the end, how these are enhancer gene interactions that you predict to be positive but aren't, and yet they're close in 1D and they're close in 3D. I'm wondering if you think that actually the Gasparini et al. Data is noisy and maybe you missed some of these interactions and that they really should be true positives, but because of noise in the training data, you think they're false positives.
Yeah, that's a good point. We thought about that because the outcome that we're predicting is whether Gasparini called it significant or not. And it's a binary classification, but they probably use a very conservative threshold to call significant. variants, right? So they want to be sure that what they call significant pairs are really significant pairs. We could go back to their data because their data also has not just the binary calls, but the actual effect sizes, and try to do like, instead of classification of regression, and see if we could predict it better that way. Yeah, I think there's definitely a possibility there. Any other questions? Okay, thank you very much.
So we're going to reconvene here at 7:45. Thanks.